So we want to start today. I will start today by acknowledging the Aboriginal and Torres Strait Islander people of the unceded lands on which we work, learn, and live. In particular, I acknowledge the Wurundjeri Woi-wurrung people, who are the traditional owners of the land where the Health Law and Ethics Network and the Melbourne Law School sit. We pay respect to elders past, present, and future, and acknowledge the importance of Indigenous knowledge in the Academy. So for this August edition of the Helen Seminar, we are doing our topic is gene therapy, unpacking where we are and where we are going. We have two speakers for you today. First up is Lorna McLeman. She is an early career clinician scientist undertaking a PhD with the University of Melbourne at St Vincent's Institute of Medical Research in the Genome Stability Unit and at the Melbourne Children's Research Institute, investigating novel gene editing techniques for Fanconi anemia patients. She is a pediatric leukemia and bone marrow transplant physician at the Royal Children's Hospital in Melbourne, and is leading a study as part of the Prepare Renew Stem Cell Medicine Consortium, investigating consumer readiness for gene therapy in Australia. Lorna has a special interest in bone marrow transplant and advanced cellular therapies, including gene therapies. After Lorna speaks, we're hearing from Katherine Lieschke. Katherine is a self-proclaimed regulatory bureaucracy nerd. Originally a biomedical scientist with an interest in immunology and microbiology, Katherine briefly detoured into secondary school teaching before returning to the scientific world to work in clinical trial startup at the Murdoch Children's Research Institute. She developed an interest in ethics of research and completed a Master of Bioethics in 2018. Katherine has since specialised in clinical trials of gene therapies, as well as the operational aspects of delivering gene therapy in a hospital context. Katherine's current project is investigating ways in which stakeholder voices can be incorporated into the Australian regulatory context for advanced therapy medicinal products. She will identify the perceived barriers and enablers to bringing cell and gene therapy to patients and utilise a guidance ethics approach to explore the ethical issues associated with these therapies. So welcome to both of our speakers, and I will now hand over to Lorna.
Thank you. Thanks so much, Chris. That's such a lovely introduction. So yes, as Chris mentioned, I'm a pediatric oncologist by training. I now work in leukemia and bone marrow transplant at the Royal Children's Hospital in Melbourne, and I have a particular interest in gene therapies and cellular therapies, particularly for those patients that are challenging to treat with our standard of care, which is a bone marrow transplant. I'm also undertaking my PhD, and we're trying to develop a novel gene editing technology for patients with Fanconi anemia, which is a cancer predisposition syndrome. But I won't be talking about that in detail today. I would love to share with you a stakeholder study, which I've been lucky enough to be the PI on, which is linked in with the new clinical gene therapy trial, which is open at the Royal Children's Hospital, the only one in Australia currently for autologous gene therapy. And that's for patients with a particular mutation called RAG1 and a condition called severe combined immune deficiency, which I'll talk to you in a little bit of detail about. We're very lucky to have this project funded through a collaborative grant through Renew, Novo Nordisk Foundation Centre for Stem Cell Medicine. So just briefly, I was unaware of the background of everybody that would be coming to today's talk. So I'll just go through the basics of DNA and how gene therapies work and how they're related to the more novel technologies of gene editing. And just a little brief background into the history, because I think it helps understand where we're up to. And I'll talk to you about the condition that we see in patients for bone marrow transplant called severe combined immune deficiency, and how we believe the opportunities for having gene therapies will be really beneficial to this particular patient group. I'll talk to you about the open trial that we have, and then also about the stakeholder study, which I'm leading. So this may be very common knowledge to many of you, but if anybody is less familiar, I just think it's always helpful to go back to the basics. So as you'll be aware, the human genome is genetic information. It's found inside a human cell, and it's mainly as DNA, and that works as chromosomes, which are inside the cell's nucleus. And all humans have 23 pairs of chromosomes. It's 46 in total. So you get 2 sets from each parent, which is 22 pairs of what's called an autosome, and 1 pair of a sex-determining chromosome, which will be either X or Y. And the proteins encoded by the cell's DNA are responsible for almost all of the work and processes required of our living cells. And variants or mutations in the DNA are responsible for about 10% of all human diseases. So these variants can be present in all cells, which include the egg and sperm, which makes them germline and inherited. Or they can be present just in the cells that are not egg and sperm, which makes them something called somatic, and it means they're not inherited from generation to generation. And technology has been developed through multiple different methods, really over a period of time, which allows us to read DNA accurately down to single nucleotide changes with a very rapid turnaround. So the Human Genome Project, which allowed us to start doing this, was only started actually in 1990, which I think is quite interesting to think back to. And it was completed in 2003. And that study mapped the entire human genome, and it cost billions of dollars to read the first human genome. But we're now very lucky to have technology, which means we're able to read an individual's genome for about $3,000. And we have different tests to look at different parts of the genes or full genomes. And by reading that genome, we can then identify areas of mutations that are responsible for genetic disease that we previously didn't even know existed. And because of this, there is much development in the understanding and treatment of many genetic diseases. So that's reading the DNA, but how we've now learned to write into the DNA is another area that I'm so lucky to be involved with. And I think it's just very exciting for our patients moving into the future. So we can now correct some of these genetic mutations by using DNA and RNA, and with technologies such as gene therapy and the newer gene editing. And it's been investigated actually for about 30 years. And it is rapidly expanding the therapeutic options that are available for individuals with genetic diseases. So to understand the rest of the talk, I just thought it was good to briefly explain what a gene therapy is. So the definition would be using genetic material for the treatment of a disease. And what that means is the therapeutic DNA or RNA that has been developed is delivered into target cells. It then needs to be transcribed and translated into the cell nucleus, if you remember your biology, and it then has the information to change a protein that's produced by target cells. These therapies can be delivered directly into patients, and that's a type of therapy we call in vivo gene therapy, and it essentially means it's a product that's injected into a person. Or ex vivo, which is what I'm more involved with as a bone marrow transplanter, by collecting the patient's cells and then correcting them outside the body before we reinfuse them into a patient. And the DNA and RNA is carried into a cell in gene therapy using a special type of envelope or a package, which we call a vector. And vectors that are very good at getting inside cells are viruses, and they work just like that envelope. And so for some conditions, this treatment may just be a once-in-a-lifetime cure as an alternative to some really risky treatments, such as a bone marrow transplant. So we may not need to go into too much detail, but I just wanted to explain to you that the manufacture and production of viral vectors is incredibly complex. So they're complex segments of DNA which are packaged together. And the aim is to deliver safely the healthy gene, which we call a transgene, efficiently into a particular target cell. They then replicate within the cells, and we also don't want them to cause excessive immune response in patients, as we will talk about a little bit later. So is it safe to use viruses? We're often asked, and we use viruses including sort of HIV for some viral vectors. And the answer to that is yes, it is safe. They are inactivated. And this means that the viral genes have been removed. So these vectors are sort of like an empty envelope. They're unable to cause infection. So just because CRISPR technology has become so prominent currently, and I think it might just be worth explaining some of the obvious differences between gene therapies and gene editing technologies, which can be described as a type of gene therapy. So gene therapies use viruses for the delivery system of DNA and RNA into cells. And they were the first discovered technology, and the first clinical trial was back in the 1990s. And that was for the immune deficiency that we'll talk about later, severe combined immune deficiency. And the vector allows this DNA or RNA to be delivered into cells, and the healthy gene or the transgene can insert randomly anywhere into a patient's genome. And that does lead to some risks, which we'll also talk about later. Gene editing, on the other hand, has been investigated in several methods previously, but it expanded dramatically in 2012 when the CRISPR-Cas9 technology was discovered through work in bacteria. And the founders went on to win a Nobel Prize, so Emmanuel Charpentier and Jennifer Doudna. And it delivers the genetic material in the same way into cells, but it doesn't use viruses. It's also really easily accessible to many researchers and has a lot less restrictions compared to gene therapy with viruses, and some potential less toxicities. But because they are newer technologies, they're still somewhat under development. There are some gene editing clinical trials, however, none for children, so none for pediatrics in Australia. Just sort of thinking back in time to give a brief idea of the timing that some of these therapies take to develop. It was back in the 70s and 80s that much of the preclinical work was done to investigate gene therapies. And the first clinical trial in patients was in 1995 in a type of severe combined immune deficiency called ADA, SCID. So this was the first condition for which autologous gene therapy was developed. And that's the gene therapy which happens outside the person's body. Then in 1992, the gene therapy for SCID was developed, which allowed us actually to not just correct immune cells, but to correct hematopoietic stem cells. What that means is it proves concept that you can genetically correct a cell that is able to repopulate the bone marrow compartment. And that's very important for many of the clinical trials and the other conditions which we treat today. In 2002, it was expanded into further genetic mutations for this particular condition. And two events that we'll return to are the development of problems with some of these therapies, including something called insertional mutagenesis, and also, very sadly, the death of a young patient in a different clinical trial because of overwhelming activation of the immune system. So gene therapies were expanded to multiple different conditions then, including metabolic disorders, and also, as you will potentially have heard about, the development of CAR T cells for treating patients with leukemia initially. It was 21 years after the first clinical trial, so it was up until 2016 that the first cellular gene therapy was granted market approval, and that was by the European Commission to GlaxoSmithKline for some Strimvelis, which is the brand name for the gene therapy product for ADA SCID. And it was granted approval under an advanced therapy medicinal product, and it was the first licensed ex vivo gene therapy product internationally. So those two issues that I said we would just discuss in a little bit further detail, which I think is important to understand when we're discussing these therapies, not only amongst professionals, but also with patients and families, are insertional mutagenesis. So this is something where in the early trials using the first generation of viral vectors for gene therapy, because of that potential for inserting randomly anywhere in a patient's genome, unfortunately, in these trials for SCID, so X1, X-linked SCID, 6 of the patients had the healthy gene insert close to an area where it activated another gene which was able to cause cancer in those patients. And very sadly, 6 patients developed T-cell acute lymphoblastic leukemia. And it could be treated in most cases, but of course, and quite rightly, it led to a halting of the development of gene therapies and led to development of second generational safer viral vectors, which are inactivated to markedly reduce this risk. Another prominent case that I mentioned is the individual up on the right of the screen. So this is 17-year-old Jesse Gelsinger. He had a metabolic condition called OTC deficiency, and he was treated with an in vivo gene therapy product using an adenoviral vector, but unfortunately had overwhelming activation of his immune system, and he passed away. But we're now much more aware of immunogenesis, as it's called, with gene therapy, and the risks and ways that we can develop products to help prevent this. So what diseases are actually treatable by gene therapy and what products are available? In children, we can broadly categorize this, and I should say I'm only talking about pediatrics because I haven't looked after adults for a very long time, is pediatric immunodeficiencies, of which there are many up on the slide there. And actually, that slide is probably a little outdated. And then hemoglobinopathies. So these are problems within the production of the red cells in the body. And that includes quite prominent conditions such as sickle cell anemia and thalassemia, and then metabolic diseases. So this is a complex group of conditions, and they're normally genetically inherited. And essentially it means there's a mutation in a metabolic pathway that leads to a buildup of a toxic substance within somebody's body, or a lack of a required enzyme. And that's normally one that's crucial to the way the body works. So you can see in the table the current open gene therapy clinical trials in Australia. The pediatric trials are in black, but you can also see there is only one current open ex vivo gene therapy clinical trial for individuals. And I'll talk to you a little bit more about this study which I'm involved with. So just briefly, I've mentioned the name several times, but just so you have an understanding of what severe combined immunodeficiency is, because it is a rare disorder and you likely wouldn't have come across it previously. It's heterogeneous, so it's caused by multiple different mutations in different genes. And it's inherited, and it results in a lack of T and B cell function often. It essentially means that patients have no functional immune system. They often also have some failure to thrive, so they don't grow very well, and it's normally diagnosed in infancy. Now we are very lucky across Australia to have newborn screening in every single state, which means that we do get to these children a little bit earlier. It is fatal universally within the first 2 years of life due to overwhelming infection without being treated. And the standard of care currently is a bone marrow transplant, which replaces the dysfunctional immune cells. However, more recently, as you've seen, gene therapy products are available for 3 different genetic mutations of severe combined immune deficiency. So why would we be interested in gene therapy when we already have the option of a bone marrow transplant? Well, actually, transplant is not without its toxicities and risks for patients. And the benefits of a gene therapy are that you do not require a donor. For many of our patients, they may not have a matched sibling donor or a matched unrelated donor. And therefore, sometimes we have to use something called a haploidentical donor, which means one of the parents. And that can increase the risks of toxicity of transplant for patients. Also in gene therapy, because you're not using cells from another person, you don't risk a condition called graft versus host disease, which causes a lot of morbidity to our patients after a transplant. And they also don't need immunosuppression because it's their own cells that they're getting back. So we can markedly reduce the risk of transplant-related complications, and generally there's felt to be lower toxicities, although we're in the early stages of these treatments. So how would we actually deliver these therapies to patients? Well, I think one of the key things here is that in vivo gene therapy, so giving the product directly to a patient, is historically more accessible to Australian patients than these cellular therapies, because we currently don't produce autologous gene therapies in Australia, which means that the process has to occur overseas and the cells moved. And that leads to a workflow that can be quite complex. And so once a patient might be eligible and selected, this is for our particular trial, the image up on the screen, they then have to undergo bone marrow priming, which means that we stimulate their stem cell production. We then have to collect those cells through a process called apheresis, and the cells have to be sent via a courier overseas. In our trial, it's to the Netherlands in Leiden. And this is where the actual transduction, so the moving of that DNA or RNA into those target cells, occurs, and the correction with the healthy gene. The cells are then frozen for the long journey back. And normally the team will preserve a small sample of cells for testing to make sure that this has been efficient. And once that's been confirmed, we then do a process called conditioning our patients. And that means that we give them chemotherapy to empty out the bone marrow and allow the new gene-corrected cells to engraft and to grow in the bone marrow space. So this is challenging to organise. There's lots of hurdles, such as local import and export licenses, organising our courier services in a timely manner. And another hurdle for us actually has been the requirement for a test sample of cells, which you can imagine in paediatrics doesn't come along that often. And when we have a patient, we need to have a patient that has excess cells that don't require them for a return themselves. So that has been a little bit of a sticking point. It's challenging when we're so far away from the centres actually doing the gene transduction. But this is the trial that we're very lucky to be involved with and open for enrolment. It's called RECOOM. So it's for patients with severe combined immunodeficiency that have a particular genetic mutation called RAG1. And it is rare, but we would expect to have about one patient diagnosed per year in Australia based on the incidence and our birth rate. And it was designed in Leiden University Medical Centre by Professor Frank Stahl and his team. And our local PIs here at the Children's Hospital are heads of transplant, Teresa Cole and Rachel Conyers. And it is non-randomized. It's a phase 1/2 trial. So that means it's investigating the safety of this therapy and also how effective it is for patients with RAG1 deficient SCID. We're the Australian site. There are multiple other sites open across Europe. And it is the first cellular gene therapy product more broadly available in Australia. Although I should mention there was one patient treated about 20 years ago in New South Wales, also on a clinical trial. So our trial inclusion and exclusion criteria, to be included in this trial, a baby must have the diagnosis. They must be between 2 months and 2 years at the time of treatment. Very, very young infants, we try not to transplant until they are over 2 months of age, and it would be the same for gene therapy because you increase the risk of complications. And these patients can't have a matched donor. And the reason for this is because we know that there are very good outcomes for patients who have a well-matched donor, either a sibling or an unrelated donor. And so ethically, these patients need to not have a match. The exclusion criteria shown up on the screen, and this includes a group of patients with something called Omen's syndrome, which you may not have heard of before, but it essentially means they have a low number of immune cells still present, so it's not completely absent, and that can actually make it a little bit more challenging in the transplant setting because these cells that are still present are dysfunctional and can cause problems. And patients can't have had a transplant before they have a gene therapy. For obvious reasons, they would then have donor cells within the bone marrow compartment. And what you're actually wanting to correct is the patient's own cells. But I think it's really important for patients and families to understand that they can have a gene therapy and then go on to have a bone marrow transplant. So this does not exclude them from the standard of care. And the trial primary endpoints are to prove feasibility of the study, to look at safety measures, and this always includes death, failure for the cells to grow within the bone marrow, so failure to engraft. If the immune cells that we're trying to grow have not sufficiently reconstituted post-gene therapy, and insertional mutagenesis, which is what I mentioned earlier about neighboring oncogenes being activated. And it's worth mentioning that I don't believe there has been one patient using this particular type of vector used in this trial that has ever had an episode of insertional mutagenesis. So these vectors are much more safe than the early gamma retroviral vectors that were used. So what happens from a patient enrollment? As I mentioned, it's a rare condition. The study's aiming to enroll 10 patients over a 2-year period. It's enrolled 3 patients internationally so far. We are open, but yet to enroll a baby. So once enrolled, as I mentioned, the patient would have their cells collected, shipped to Leiden, transduced with the product, and then only when we have proved that that it's been an efficient transduction would we condition the patient and so give them the chemotherapy that empties out the bone marrow. And then the product's released and given back. And there is up to a 15-year follow-up per patient, which is actually much longer than most clinical trials, and that's standard of care for gene therapy trials. So there's been significant regulatory steps to initiate this study, including establishing a workflow in this novel process. And my colleague, Dr. Hannah Walker, was responsible for much of the groundwork here. And it included gaining trial sponsorship from Melbourne Clinical Trials Centre, for applying for safety and ethical approval, which can actually occur at any Australian site, despite the fact that the trial's occurring in the Royal Children's Hospital. And this ethics was passed in Sydney. For local governance sign-off, and for developing standard of care procedures within our hospital for the trial itself and all associated procedural steps. So that includes all the way from apheresing the patient's cells to how to dispose of the gene therapy clinical waste. and also the stakeholder education and engagement, which is part of the trial that my study is also included in. So just to mention that we are funded through Renew, and many people, I guess, would be unaware of Renew. It's a huge international consortium, and it actually links Leiden University Medical Centre, which is where this product is developed and made, the University of Copenhagen, and MCRI here in Melbourne. And the aim of Renew is to bring together international scientists and clinicians to advance stem cell medicine, including gene therapies, and bring discoveries in a more expedited way from preclinical to clinical work. And the Prepare team within this bridge the gap between the clinical and scientific community to enhance the understanding of our patients and families and our healthcare professional colleagues, because these are the groups of people that really matter the most to us. So we're at this really key time point in Australia regarding access, involvement, and future development of cellular gene therapies and eventually gene editing, hopefully, in clinical trials. And as more gene therapy trials become accessible locally for our patients, we want to better understand what bioethical factors might impact involvement and enrolment on trial, and how we can best educate our families and staff to make informed decisions regarding these therapies. So our study sits under the Prepare theme of Renew, and it's targeted towards engaging our current and future stakeholders to improve representation, as well as identifying hopes and concerns of key groups at this unique time point. So you can imagine there may be significant barriers to implementing these therapies. And some of these include the huge funding costs of these individualized treatments and ensuring there's equity of access across socioeconomic, ethnic, and cultural backgrounds in Australia. We want to engage the community in education around novel therapies and to enhance the informed consent process and decision-making, because they are complex therapies to understand. And we want clinician and consumers' understanding to be really good of the risk-benefit ratios of these therapies compared to the standard of care when they're making these decisions. There are some issues, as I mentioned when going through the history, in the time that it takes for a product to be produced in a preclinical setting and to actually get to patient access. And how will these novel gene editing technologies challenge our current regulatory system? Katherine will talk in some more detail, but partly because of the individualized nature of some of the newer therapies, this is really going to challenge our regulation system, and it's likely to need to adapt to keep up. And the development of infrastructure here in Australia is critically important for the delivery and eventual local manufacture of these therapies, which will mean that our people in Australia will have more availability and more access to trials. We also need a very highly skilled and trained workforce for all different areas of delivering these products. So as you can see from thinking more, changing tack to think about the education and engagement perspective, some of the complexities of these therapies can lead to real challenges in consenting and decision-making. And there's a lot of information publicly available from many media sources, and some of this is evidence-based and some of it not so much evidence-based. And so we want to think about, in our communication and engagement with families and the community, how can we best educate them to make informed consent decisions, and how can we communicate the long-term risk and benefit ratios to them in some of these newer therapies, and think about how gene therapy might affect the quality of life for families compared to our standard of care, and in my role that would normally be a bone marrow transplant. And for healthcare professionals, how can we support our colleagues in educating families around a therapy that they may not have huge amounts of experience in themselves? And how can we balance any conflicts of interest, as many people have deep interest in gene therapy clinical trials, and it doesn't have to be financial. The pressures for success of early phase clinical trials are present. So the project that I'm involved with has some aims, and it's important to understand that there's a real paucity in the data in the literature of any data in this area. So there is a gap to be filled, and we want to map the views, beliefs, and concerns in particular key stakeholder groups in our study in two arms. So we have parents of individuals with severe combined immune deficiency who have been so generous to be involved with our study from the beginning. as well as via the two consumer advocate groups for immunodeficiencies in Australia, OzPIPS and IDFA. And also healthcare professionals. So we want that to be people that are strongly involved in looking after these patients, but are spread across bone marrow transplant physicians and immunologists, nurses, allied health, and laboratory scientists that would be looking after these therapies when they return to us. And it's very interesting, but we don't just want to document these viewpoints. We want this to come full circle and use the data we generate to implement change in future clinical trial design. And we also plan to use our data endpoints to develop an educational package around gene therapy that's effective, but also addresses the needs of families that could be specific to our particular trial for RAG1 severe combined immune deficiency, but also adaptable to future gene therapy or even editing clinical trials. These are our projected outcomes of our trial. We're currently enrolling in both arms of our study and performing semi-structured interviews with some key and very generous stakeholders that have given their time to be involved. And we do want to map these viewpoints, but also identify key areas of concern or potential hurdles so that we can adapt future clinical trial design based on the data that we generate. And we aim to resolve the misinformation that's out there with our modifiable educational package for families. And we're also, which is, I think, a really key point, interested in developing a national network of like-minded individuals who are interested in advancing these therapies responsibly within Australia and New Zealand. So with that, I'll hand over to Katherine, who will talk to you a little bit more about the regulatory side of things. But I wanted to thank Renew for funding our study, our Prepare group, and our RAG1 SCID clinical trial team. So thanks so much.
All right, let's go. A crash course in the regulation of clinical trials of gene therapies and some of the ethical issues around them. So Chris gave me an introduction, and I don't really need to say anything more, except that my current role is twofold. I am the Gene Therapy Trials Readiness Coordinator at the Melbourne Children Trials Centre, which is definitely a made-up role. That's not a real thing. And I'm also a PhD student looking more broadly at advanced therapies and the regulation of them. Most of my experience is in vivo gene therapy. I'll remind you shortly of what that is compared to what Lorna's been talking about. But I'm going to try and be clear where I'm talking about something that's specific to one kind of gene therapy and something that's more broadly applicable. I'm going to talk to you about how clinical trials of gene therapies are regulated. I'm going to talk about some of the challenges that we're finding in the regulation of these trials as we're starting to do them, and also highlight a few of the ethical considerations around clinical trials of gene therapies. I'm going to focus on trials because that's where this is largely being done at the moment. We aren't doing a ton of this outside of clinical trials, so we're going to focus on that context. Quick reminder that we have two types of gene therapy. We have the ex vivo, where we change the patient's cells outside of the patient, and then we have in vivo, where we're putting the vector directly into the patient to change them, the genetic information inside them. This is where we're putting viruses into patients, and sometimes that can sound pretty scary. And this is, I think, where people start to have questions around who's checking what you're doing. So these are the people who at least check what we're doing in every single clinical trial. So gene therapy clinical trials are regulated in the same way as any clinical trial in Australia is. And we regulate clinical trials for two main reasons: to protect our participants from exploitation and unnecessary harm, and to ensure that the results of the research are trustworthy, that is, that the results are valid. So the Therapeutic Goods Administration, the TGA, regulates all clinical trials and checks that the safety of the patients has been adequately considered. They look at the safety data of the product up to that point. They also look at the design of the trial and say, yes, this looks like a trial that can get us answers, that can get us results that we can use. Ethics committees are involved in the ethical review of the trial, and they review everything that we give to patients. So particularly they're looking at protecting our patients' rights and our patients' safety, but also all of the information that we give our patients to help them make decisions about whether or not they want to participate in a clinical trial. Research governance is the part of the review that is about can we do this particular location? Can we actually do what we say we're going to do? Do we have the money? Do we have the space? Do we have the people? And also, do we have people who are checking that all the right things are being done? But when we have a gene therapy product that involves a genetically modified organism, like a virus, and this is something that has been modified, it's an animal, plant, or other organism, like a virus, that has been modified using gene technology. And when we have one of these GMOs, we've got additional regulation. So this is particularly important when we're thinking about in vivo gene therapy products, but it can apply to many different kinds of gene therapy. Then we have two more players coming into the space. We have the Office of the Gene Technology Regulator. This is a government department that reviews the product and planned activities with that product to check the biosafety and containment of the organism has been adequately considered in the plan. Different products are going to require different levels of containment, but the OGTR checks that the plan that we have to handle the product is safe for the people who are using it and for the general public. They can also monitor and inspect the work we're doing to confirm that we're doing it correctly and as we said we would. They don't look specifically at patient safety in that traditional sense, as that's the remit of the TGA. Now, unless it's an exempt dealing, work with GMOs is going to require a license from the OGTR. The exact definition of an exempt dealing is a bunch of things in a list in the regulation. But CAR T cells, for example, and products like the RAG1 SCID therapy that Lorna spoke about are exempt dealings. But a lot of the gene therapies that I've worked with require these licenses because they are viral vectors that we are handling on site. So the Institutional Biosafety Committee is the people who at the site check that we are going to be compliant with the OGTR's rules. They give us advice on our work practices and the way we can keep ourselves safe here on site. So we're talking about genetically modified organisms, but the thing that I think is really important to take away is not all gene therapy products are going to need OGTR review. It's very tempting to see the word gene therapy and start that OGTR application process, but it doesn't apply to everything. It only applies to GMOs. So why do we regulate these GMO products specifically rather than differently from other medicines? Well, it's because there are some additional risks posed by them, or specific risks posed by them that we're interested in. Now, when these risks are handled, when these products are handled appropriately, the risks are small. But there are risks to healthcare workers, the public, and to patients if these products were just, I don't know, let out into the world. I think of this as differently risky rather than more risky, but we use the same principles we use for any risk in healthcare. We contain it and we manage that risk. There are different levels of risk that correspond with these 4 biosecurity levels. You don't need to know what makes something go higher, but basically higher, worse. The risk level is based on the characteristics of the GMO in question. I want to be clear that GMO medicines are not more dangerous intrinsically than anything that we use on a regular basis in a hospital. There are many, many dangerous medicines that we use for specific purposes. Also, it's important to note that humans who have been treated with gene therapy are not considered GMOs for Australian regulatory purposes. It's a little counterintuitive, but it does make doing things a little bit easier. So gene and cell therapies are emerging fields of medicine, and regulating new fields is hard for a number of reasons. Firstly, existing systems for review and approval might not be fit for purpose. For example, forms might not have relevant questions, or they might ask questions that are really just inappropriate to ask in this context. So you're trying to fit square pegs into round holes a lot of the time. The system itself will have inefficiencies, and we need coordination between different groups of people who perhaps traditionally have never spoken to each other in order to establish an effective and efficient system where we don't have duplication of effort and we've streamlined as much as possible. And this is really important when we find out about new things that we need to consider that we haven't thought about before. Existing processes for oversight might not be feasible or effective in a new context, so we need to make sure that those work. And regulation changes really slowly, while discovery can move quite quickly. So we've been lucky in this space that the OGTR in particular have been very good about working with us to work out how to regulate these products. But regulation, especially when it's enshrined in law, takes a long time to change, while science is not required to go through sort of the same deliberative process before it comes up with new information. So what is it about gene therapy trials? And I'm going to include gene-modified cell therapies in this that makes them a little bit different. Well, as Lorna mentioned, there's high hopes for gene therapies. They are theoretically curative, and therefore the risk-benefit ratio is somewhat different than the more traditional therapies for these kinds of conditions. We're talking about single one-time dosing with the product, which means that studies look a bit different to what we're used to. We don't like when things look different. And also there's a permanency of the physiological change that we're making in the patient that we need to consider in the design of our studies. Follow-up is pretty intense. We're often requiring patients to stay in hospital for a week and then to stay around the hospital for a month, but also do long periods of follow-up on a clinical trial, 15 years in Australia, but there's talk of whether or not we need lifelong follow-up for people treated with these gene therapies because they're permanent. And this starts to get even more complicated when we think about, now we've got some ethical considerations as well. These are not just complicated therapies from a technical standpoint, they're ethically complex as well. We have limited human data to use to predict the results of this therapy. We can't predict the side effects because we just don't have much data. We know there are immune responses to the therapy, especially vectors in vivo gene therapy, that can limit future treatment options, even if a therapy enters standard practice. You can end up being excluded from other experimental treatments, or from future approved therapies because of the permanent effects gene therapy can have on the body. These are not yet known or understood in all cases. And because we've got a permanent change in the body, any side effects from the treatment may not be able to be resolved, particularly in vivo therapy where the cell type that's being changed is much less controlled. It's not possible to unedit the cells if the change proves ineffective or problematic. And Lorna alluded to some of the problems of patient expectation and consent. And I don't want to dwell on this too much because I think Lorna spoke really well about it. But decision-making is tricky when you don't have all of the information, which you never do in a clinical trial context. But gene and cell therapies are really complex and they're hard to explain to lay people. And we're dealing with a therapy type that's had a lot of hype in the media. A lot of people have been exposed to misinformation, outdated information, or have an incomplete understanding. So we're working through and against these issues when we're trying to communicate with people. We've also got a pressure to make a decision quickly in this context. And I just wanted to give an example of this because many of these gene and cell therapies are more effective if they're given earlier, and even days can matter in these cases. In spinal muscular atrophy, where we do have an approved gene therapy, the gene therapy Zolgensma is available for babies under 9 months. But if the children are diagnosed late, then the window might be small before they age out of the therapy. But the clinician at RCH who works with these patients describes it as a disease where time is neurons. Every day you don't treat, the child is getting sicker. But he also reports that it takes up to 5 conversations with the families for the risks of gene therapy to be properly understood. There's a clear tension between the urgency to act and the necessity of time for adequate discussion and informed decision-making. The stuff I have on this slide are not the kinds of concerns that are unique to gene therapies. These come up all the time in lots of types of clinical trials, but gene therapies sort of have this way of highlighting them and bringing them to our attention in a new way. Placebos are problematic with gene therapies because designing an appropriate placebo is hard. If the placebo includes the vector, then immune response to the vector can be really problematic and potentially harmful, but also restrict future treatment options. Delivery route for gene therapy can also be risky. We're now looking at trials where we're directly injecting gene therapy into the brain of babies. You don't want to do that with a placebo because that's a very risky procedure for no potential benefit. We know these are high burden on participants and their families with lengthy treatment periods and long follow-up. And the question of who gets to have a go, who gets to try these new therapies, this cool new technology, this potential curative treatment is a big issue. Trials are often restricted to the sickest patients, but given gene therapy is more effective earlier in disease progression, we need to consider where the appropriate point to intervene and to test an intervention might be. This ties into issues around, do we even have a way of measuring what a meaningful change is? And have we even spoken to people to find out what a meaningful change might be for them? it might not be what we expect. As a pediatric institution, we tend to not ask so often, can we do this or should we do this in children, but rather, can we do this in children? But because gene therapy works best if delivered early, we need to look at administering gene therapies in childhood, perhaps not just in conditions where the condition is lethal in childhood. We may be looking at intervening, even though the child will grow into an adult, and we could ask them then. We may need to look at intervening earlier. And translation of trial results is a massive problem. Trials are very artificial and things don't necessarily translate super well to the broader population. We also have system issues. We need an equitable system. We don't want to exacerbate existing inequalities. The system has to be able to deal with a certain amount of stuff we don't know, and that often breaks a system. So we need to look at what might be at play here and how can we modify our system or make our system adaptive to what we do know. If these therapies work, they're very hard to implement in a healthcare system. They're resource intensive and they're difficult to do. We need national coordination. We need proactive readiness to deliver these therapies, and that should really be a priority for Australia. Sustainability is a massive problem too. These are very expensive. We need to have difficult discussions about how we're going to decide who will pay and what we'll pay for. We have some systems for determining value in healthcare already, but we need to consider whether these systems are actually fit for this purpose. So where to next? This is where Prepare comes in. We try and solve some of these problems before they're massive issues. We're a multidisciplinary team and we're exploring questions of how we can make the world ready for these therapies. As Chris mentioned, my work looks at how we can support the system that's making decisions about these medicines. And I'm exploring a guidance ethics approach, which is a methodology developed in the Netherlands. Up until now, it's been used to look at digital technologies in various contexts, but I'm going to try and do it with an advanced therapy and see what happens. My conclusion is we need to be prepared, but not alarmed. I'm really proud of that. And to acknowledge the amazing stem cell ethics and policy team that I work within, my PhD supervisors, Professor Megan Munsey, Dr. Chris Gingell, and Professor Martine de Vries. The Melbourne Children's Trials Centre and the MCRI Genetic Health Group have been instrumental in creating the gene therapy readiness position here at MCRI and in funding that. The MCRI IBC, the RCH Research Ethics and Governance Office, who answer all my questions when I wander over and go, so am I allowed to do this? And Sydney Children's Hospital Network and my colleagues there who have let us learn so much from their own experience of doing things and have also been really great in discussing how we might make national networks to help solve these problems. And on that note, we are finished and I'm going to stop sharing so we can have a discussion and get Lorna back in frame.
Thank you, guys. That was amazing. Great, perfect. Okay, well, look, thank you both again. That was a really fascinating talk. Everyone, please thank our speakers, Lorna and Katherine. And please look out for the emails for our September Helen seminar. And if you want to join the Helen mailing list, if you're not already on it, we've just put a link into the chat there. So thank you very much, everyone, and have a great afternoon.